So after interphase comes something called mitosis. Mitosis is also called karyokinesis. Karyo, breaking that word down into two parts. Karyo means nucleus, and kinesis means to split. Kinesis means to split. And so as we're looking at this, the nucleus is dividing or splitting, and this includes, or mitosis includes, four types, and then what we call it is PMAT. Very easy way to remember it. Interphase is always first, and interphase goes before the P, and then we've got PMAT, and PMAT includes all of our mitosis. So we start with the P, and what P represents is something called prophase. In prophase, there are a couple of things that happen. First of all, what happens is the nuclear membrane kind of goes away, and this chromatin condenses into chromosomes. Hopefully you recognize right over here, this says, what is that? You recognize that this is a type of our DNA, and that type is called a chromatin. It condenses into chromosomes. So this long stringy stuff begins shrinking and shrinking and shrinking until it looks something like this, or a chromosome. Whenever it begins shrinking, what it then happens is you get that one chromosome becoming two chromatids made up of that DNA that's replicated in the S phase. So two chromatids being one side is over here and the second side is over on this side. So now it's one full chromosome. And the last thing that happens here in this prophase is we've got this chromatid, one of these chromatids, being duplicated. So now we call these identical sister chromatids. The last thing that happens here is the centrioles that are sitting in the middle of our cell and that are only in animal cells begin moving to opposite sides of the cell. And they begin forming these fibers, which are these little lines over here. They begin forming these fibers, and they start attaching those fibers to the center of a chromosome, which is called, right here in the middle, center of a chromosome, which is called a centromere. A lot of definitions in this. So a centromere, which is sitting in the middle of that. So that's what prophase is and what it looks like. And so in our full little view here, let me go back to this full little view here. What we can see is that it looks something like this. Our nuclear envelope here has faded. Our chromosome, we can tell, have been condensed. So we start seeing little gaps. This is not a black dot anymore. We start seeing gaps in here, and we start seeing chromosomes or X's. And then these centrioles out here move to opposite poles, and those fibers begin reaching out here. And so if you see a cell that looks a little bit like that, you can tell that it is in prophase. Moving on to our next one, I said that's PMAT. Next one is M. M is metaphase, and this one's a very easy one. Metaphase standing for middle. All of those chromosomes line up in the middle of my cell. And this one right here is our full cell all the way on the outside over here. And right here, all of these chromosomes are lining up in the middle there. So what's happening in metaphase? Metaphase is where tension begins forming in the middle of at those spindle fibers, and it ends up pulling on each other. And so we've got a cell sitting right here. We've got those centrioles that I usually look like this forming on this side, and they've got those spindle fibers coming out, and they're attaching to the centromere or the center of a chromosome that have been sitting in the middle over here. And when they attach to the center of the chromosome, they just begin pulling on these chromosomes. And when they begin pulling, it ends up just putting them very easily in the middle. As you pull something, if you're pulling with equal weight, it's just going to end up in the very middle. So that's what happens. The cellular equator, is also called, what we call this is the chromosomes line up on the metaphase plate. Very easy. In the middle on the metaphase plate. That's what we call that. So our next one, after that, this is what the cells usually look like. The next one after P, M is A. PMAT after M is A, and that's called anaphase. Three things are happening during this time. One of those is that the spindle fibers begin to shorten, which means that they start pulling on these chromosomes. Another thing is that those chromosomes actually begin getting split in half at the centromere and are pulled to the opposite side. So what we got here is these chromosomes were sitting as X's in the middle, and then they got split in half and are pulled to the opposite sides. So now no longer is it a chromosome sitting there. On each side, a chromatid is being pulled to the opposite side. A chromatid is being pulled to this side and that side, and they're being pulled to opposite poles of the cell. Usually, it looks like A's in there, so that's how I tell this is anaphase. This anaphase ends when there are two daughter chromosomes located on the opposite sides of the cell. All right, and then lastly, the last part of mitosis is called telophase. In telophase, we are now on this T of PMAT. In telophase, the spindle fibers begin to disappear. So those fibers coming from these centrioles begin to disappear. And lastly, the nuclear membrane of each side now begins to reform. And so you can tell right over here we've got a little jagged line forming around these daughter chromosomes in here. We've got another side forming over here. And not quite is this cell split in half yet. And that is the end of mitosis. So this is what a kind of a real picture would look like. You can tell that all of these, there's now a black dot forming in here, and it's on the either side, and you can tell that there's no real split between this cell. And so, so the last part of our cell division is called cytokinesis. This is not part of mitosis. This is the very last part of the whole cell division process. And cyto is talking about the cytoplasm. Kinesis is talking about the cytoplasm splitting. And so what we would get here in the cytokinesis is once the nucleus has been divided, once it's separated on either side, the cytoplasm also needs to divide. And so what we have here is two different types. In a plant cell is squared, and an animal cell, like your cheek, is rounded. And to split these two in half, after they're split, we've got two different things. One of them in a plant, when it begins to split, is called a cell plate. It forms right down the middle of it. I'll use that as a dotted line. And that cell plate is just like a indentation right in between it after it's been split. You've got nucleus over here, and it's filled in with DNA, and you got nucleus over here, and it's filled in with DNA because it's already went through the process of mitosis. Over here, what we have is something called a cleavage furrow in animal cells. And what happens is this side will begin to indent just like that, and this side will begin to indent just like that, and sooner or later it just pinches itself off, and that's called a cleavage furrow. And that's cytokinesis. So what we've got here is this whole process of cell division. Now, there are some things that can happen, and a lot of you, I'm sure, have had family members with cancer, and it's a very, very sad, bad thing as well. But during cell division, cancer cells form when they are uncontrolled. And so cells just begin replicating and making more of themselves, and then a tumor begins forming. And when I say that they're uncontrolled because during all of this phase that we have here, this big circle, we've got interphase on the outside, we've got this small portion of mitosis, and then we've got cytokinesis in there as well. That's my little diagram of a pie chart. Each one has a Each spot has a checkpoint. We've got the G1 up here, we've got the S phase here, and we've got the G2 here. Each spot has a checkpoint. Well, when cancer cells are formed, cancer cells just uncontrollably go through this cell division process, and they go through without stopping at those checkpoints. And that's when more cells and more cells are made, and that's when a tumor gets formed. So it just continuously goes without going through those checkpoints. And so a couple things that can happen with cancer cells, but during cell division, this part you're going to write down here on your notes. Cancer cells are uncontrolled. They form lumps called tumors. Benign tumors remain clustered and they stay in one spot and they can be removed. And then we've got the other one, if you remember, it's called a malignant tumor. And that's when they're metastasized or they break off, they break away, and they can form more tumors elsewhere in the body. And those are obviously really bad. So, standard cancer treatments like chemotherapy, they usually kill both the cancer cells and your healthy cells, which was why chemotherapy is not always the best thing. Some things that can form cancer are these things called carcinogens, like tanning beds, cancer sticks is what I call them, or cigarettes. And then this one is just a charred steak, which is just kind of like a little joke there, but it's a myth. And that's mitosis.